Hello and welcome to part 3 of this introductory discussion on chemistry. This lecture is meant for my students in my microbiology course that I teach. But of course, anyone who's getting into chemistry or wants to learn a little bit about chemistry and biochemistry, you can jump aboard and enjoy this lecture. If you watched part 2, I want to apologize. At the end of part 2, my cat was outside my bedroom door here and was meowing and distracting me quite a bit. So I said a couple of things backwards, and if I seemed a little bit distracted, that's why. I had an annoying cat out there meowing, and I didn't want to stop and start recording again. So anyway, the cat's taken care of. I mean that in a nice way. He wanted to go outside, and so he's outside frolicking out in the woods right now. So hopefully no interruptions this time. So we were in the middle of looking at the macromolecules, the 4 macromolecules, sometimes called biomolecules, and the first one we looked at was carbohydrates. Now we're going to look at lipids. Lipids, aka fats, so you could say, these are lipids make up the primary components of cell membranes. And if you see in the picture down below the representation of the cell membrane, you have the yellow spherical circles there that are on the top and the bottom. Those are the phosphate groups, and the green squigglies in between, those are the lipids, and they make up a majority of the cell membrane. We'll talk more about this when we look specifically at the cell membrane in another chapter, a couple of chapters from now, looking at the cell. They consist of carbon, hydrogen, and oxygen, just like carbohydrates do, so the same 3 elements. And lipids are nonpolar and therefore insoluble in water. It's essential for life. Cells won't dissolve as a result of this because lipids are making up a large portion of the cell membrane, and cells are bathed in liquid solutions. They have water on the inside of them. They have water on the outside of them. And so therefore they would, if cell membranes were made of polar compounds, they would dissolve. But cell membranes are primarily made of nonpolar lipids, and so therefore they won't dissolve in the water that they are bathed in, and the water from the inside will not dissolve the inside of the cell membrane either. So this works out great for a cell, and it wraps around on itself and forms an enclosed structure in which cytoplasm and organelles can be found. A nucleus if it's a eukaryotic cell, or just housing cytoplasm and ribosomes and some nucleic acids in the case of bacteria. So lipids are essential components of them. What do they look like? So this is a picture of one that shows, in this example, a triglyceride. The monomers, or the building blocks of lipids, are glycerol and fatty acids, and this is a fact that I want my micro students to know. I want them to know the building blocks, the monomers of these 4 macromolecules, and I want them to know the polymers. So the monomers are glycerols and fatty acids, and the polymers then of course are lipids. So these can be formed by dehydration synthesis. Unsaturated and saturated fats. So unsaturated fats have 1 or more double bonds. Saturated fats have only single bonds. They're saturated with hydrogen, and that's why they're called saturated fats. So if you look at this triglyceride here that is in the example, 2 of its glycerides are made of saturated fat. They're saturated with hydrogen. And then the third one is an unsaturated chain, and so it has a double bond in there. So a couple of hydrogens are removed, a double bond is incorporated into the molecule, and now we have an unsaturated glyceride. So the glycerol part of this lipid would be on the left in the yellow region. That's the glycerol part. And then the fatty acids are the chains that are coming off. So you have saturated fatty acid chains, and you have the unsaturated fatty acid chain. And that's really what makes a difference. Now we talk about it in terms of health and physical characteristics and so forth, but let's just stick to why they're called saturated and why they're called unsaturated. Saturated have only single bonds. They have the maximum number of hydrogens that can exist within that fatty acid chain. Unsaturated fats have at least 1 double bond, and that causes a kink in that fatty acid chain. they can be complex lipids. So you've got the glycerol and the fatty acid chain, but then attached to that is something else. Like, for example, a phospholipid in the cell membrane. So you have a phosphate group that is attached to the glycerol, which is attached to the fatty acid chains. And so we call them complex lipids. Steroids would be a complex lipid as well. They consist of 4 carbon rings with an OH group attached to one of the rings. As you can see in this example of cholesterol, which is a steroid, the OH group is attached at the A ring of the steroid. So we have the 4 carbon rings, A, B, C, D, and with the OH group attached to one of those rings. Sterols, which is this group of compounds that we're looking at here, they're part of cell membranes. They keep them fluid at low temperatures. So they move through the phospholipid bilayer, continually moving through it, and they enable the cell, they enable the cell membrane to stay a very fluid structure because it needs to be. Things have to be exported out of the cell, imported into the cell. Proteins have to move, rearrange themselves, twist and turn, and so the membrane needs to be a fluid structure. So some examples of steroids are cholesterol, estrogen, testosterone, progesterone, bile salts, and others as well. We also call them hormones too. As you know, estrogen, of course, and testosterone and progesterone are all hormones. Well, they're steroids, which are a class of lipid-based compounds. Our third group is proteins. So we have carbohydrates, lipids, and now the third macromolecule is protein. Proteins are essential in cell structure and function. That's like the understatement of the year. Proteins are doing everything. The DNA, I mean, it contains instructions to make nothing but proteins. And so what all life basically is a manifestation of the nucleic acids. And what is that manifestation? Proteins. And so proteins are what are being produced that control our height, our hair color, our skin color, even our behaviors. In some cases, scientists even will say our personalities. The structure of an organism. Does it grow feathers? Does it grow skin? Does it grow fur? And so forth. Proteins are all the basis of all of these functions that are going on within organisms and giving them their characteristics as well. Enzymes are an excellent example of a protein. They increase the rate of chemical reactions by lowering the activation energy, and they're essential to all life forms. Transporter proteins move chemicals in and out of membranes. Flagella are made of proteins. Some bacterial toxins are proteins. They consist of monomers called amino acids. So the building blocks of proteins are amino acids. And they're an acid, so they have a low pH. And so this is involved in the chemistry of proteins. So monomers, amino acids, and the polymer then would be a protein. How are they held together? They're held together by peptide bonds. They join amino acids together, and the amino acids then are joined together by dehydration synthesis. So you have the 2 amino acids on the left there, glycine and alanine. Remove water, and so now you have dehydration synthesis taking place, and a peptide bond forms, and we have glycosalanine, which is a dipeptide, and then water as a byproduct of that reaction. So these are special covalent bonds that form between amino acids called peptide bonds. If you remember in the introductory chapter, I mentioned a compound that forms the cell walls of bacteria called peptidoglycan. Peptid, so that's referring to the protein chains that are within that compound that make up bacterial cell walls. There are 4 levels of protein structure, and each level adds to the complexity and functionality of the protein. And so this is what makes protein such a diverse category, the most diverse category of the macromolecules, is that they have levels of structure, and then other macromolecules can actually join with them. So you can have glycoproteins, for example, where carbohydrates join with proteins. And so this just adds another layer of functionality and complexity to proteins. But beginning at the initial level, it's called the primary structure. It's just a polypeptide chain. It's just one amino acid joined to another amino acid joined to another amino acid. And in this diagram here of this polypeptide chain, you notice that there are those R's that are highlighted in green. Those are what are called the R groups, and the R groups are different for each amino acid. So there are 20 amino acids that are utilized in living things. And each one of those amino acids has a different R group. The carbon skeleton is basically the same, but there's an R group that differentiates one amino acid from another. And so that is what basically makes the protein alphabet, so to speak. Each amino acid is like a letter in the protein alphabet. And what makes each amino acid different? It's the R group. That's what makes one of them look different, has a different element in it, has a slightly different functionality to it. And so this is what DNA is calling. It's calling for each of these amino acids to be joined together at the ribosome through the process of translation. One amino acid comes in, another amino acid comes in, and it gets built up. And depending on what amino acids are making up the protein, that will dictate what the protein's function is. So this is the initial level, the primary structure level. The secondary structure occurs when the amino acid chain folds and coils into a regular helix, or what are called pleats. They're called pleated sheets, like like on your bed, you got the pleated corners that go around the corner of your mattress. So these are this is giving a shape to the protein. It's giving it three-dimensionality, which is what makes life three-dimensional. This is what's contributing to it. Now, as we get up in levels of protein structure, we get some depth and dimension to the protein. And with that, we get additional levels of functionality as well. The tertiary structure is the third level of structure. We're increasing in complexity and in the ability of the protein to function. And so now we have helical coils, we have pleated sheets, we have chains of polypeptides and so forth, all joined together. But now, instead of being just a straight coil or just a row of pleated sheets or just a straight chain, these at the tertiary level, they get twisted around, they get bent, and they get put into a specific shape. And then that shape is held together by what are called disulfide bridges, where you have the element sulfur now is involved in holding these proteins together into their very specific conformations. And so this gives more dimensionality to it, and it increases its levels of function as well. You maybe have, say, when you would have smelled this, for example, like if you're straightening your hair and you leave the hair straightener in too long or not too long, you still smell that. It's kind of like that sulfur, almost like a rotten egg smell. Well, what's happening is when you're straightening your hair, you are breaking the disulfide bridges that are holding the keratin together in your hair. And keratin is at the tertiary level. And so as a result, the keratin then unravels because the disulfide bridges break, and then the sulfur that is being released from the break of these disulfide bridges combines with the moisture that's in the air or the water that's in your hair, and it creates that hydrogen sulfide smell, that kind of eggy, rotten egg smell. It's not very pleasant, I guess. I don't know. I don't straighten my hair, so I don't know firsthand, but I have smelled it. Like, for example, when my wife straightens her hair, you can kind of get that, you get that odor, that smell. And so what's happening is the high temperature is breaking those disulfide bridges in the keratin and then creating that aroma. So just I don't know. Think about that next time. If you're straightening your hair, if that's what you do, think about that. When you're doing that, you're breaking disulfide bridges, straightening out the keratin, which then straightens the overall appearance of your hair. Isn't that interesting? The quaternary structure level is the fourth level of structure. And look at the beginning of a QUAT, quat, and so that's the fourth level of structure. Like a quarter, for example. A fourth of a dollar. So we're at the fourth level. This consists of two or more complexly folded polypeptides joined together. So it's not like it's increasing its complex shape or anything like that. It's just taking maybe two or three tertiary structure proteins and joining them together, or a tertiary and a secondary, or something like that, complexly folded ones, joining them together and creating an even higher level of complexity and functionality. Conjugated proteins, to conjugate something means to come together, they consist of amino acids and other organic molecules. Like I mentioned before, glycoproteins contain sugars, carbohydrates. Nucleoproteins have nucleic acids attached to them as well. And lipoproteins contain lipids. So these are proteins and lipids joined together. And so now you have the function of a protein joined together with a carbohydrate. So maybe you have a protein that's serving with having structural purposes in some way. Or it's carrying nucleic acids on it as well to enable transport into a cell, for example, or movement through a cell. And so this is giving more levels of complexity and functionality because they're joining together. And how many times can I say levels of complexity and functionality? I mean, this is what it's all about with proteins. They're complex and they have a large amount of functions. And the reason why is from what I just explained. Multiple levels of structure joining with other compounds, and it's crazy. They're just doing so much. They're carrying out like 99% of all the function within a cell. All can be based on proteins in some way. And the last macromolecule we'll look at are the nucleic acids. Their monomers, or their building blocks, are called nucleotides. So the monomers are nucleotides, the polymers then would be nucleic acids. Nucleotides consist then of three basic parts: a 5-carbon sugar, a pentose, of one or the other, either ribose or deoxyribose, depending if it's RNA or DNA, a phosphate group, and a nitrogen-containing base. And altogether, there are 5 nitrogenous bases. There's adenine and thymine, there's guanine and cytosine, and there's uracil. And these are the basic building blocks of RNA and DNA. So the sugar phosphates make up the backbone of the DNA, and then the nitrogenous bases make up what you could say would be like the rungs of the ladder. The nitrogenous bases are joining together through double or triple covalent bonds. And as you can see in this picture, adenine and thymine are joined together by double hydrogen bonds, and guanine and cytosine are joined together by triple hydrogen bonds. So what's the difference? I kind of explained that very basically. DNA, differences between DNA and RNA. DNA has deoxyribose. It has adenine and thymine bonding together, and cytosine and guanine joining together. It exists as a double helix. So imagine it's like a closed book. All the information is on the inside and the sugar phosphates are on the outside, keeping all of the precious information locked inside the double helix. This structure was first elucidated by Watson and Crick in 1953. There was a great race among the scientists of the world at that time, mainly in the United States and Europe, to discover and describe and elucidate the structure of DNA. Many scientists were making it a much more complex molecule than it actually is. Watson and Crick kind of stuck to a simple hypothesis to its structure, and they won the race. 1953, they won the Nobel Prize as well for it, along with Maurice Wilkins as well. Rosalind Franklin was involved in this as well. She was instrumental. She was not a winner of the Nobel Prize because she unfortunately had died before the Nobel Prize was awarded, and they do not award Nobel Prizes posthumously. But without Rosalind Franklin, I don't know if Watson and Crick would have been able to discover that. But sorry, getting off topic. It's a fascinating story about the discovery of DNA, and I like to talk about it, but I'm going to stop now. Sorry. RNA, ribonucleic acid, so it has the sugar ribose instead of deoxyribose. It's single-stranded, not a double helix. Adenine now bonds with uracil, not thymine, and cytosine bonds with guanine, just like it does in DNA. So everything about its structure is the same, except we have ribose instead of deoxyribose. It's an open book. It's a single helix or a single strand in which the nitrogenous bases are open and exposed and able to be read by ribosomes. In the form of DNA, it can't be read. Imagine it's like a closed book. Instead of thymine, there's uracil, adenine binding with uracil instead of thymine. And there are some hypotheses out there as to why thymine has to leave and uracil replaces it, but we don't need to get into that discussion. Here's a good video on DNA to protein. It's a pretty cool animation of how this happens through the process of transcription and translation, and it shows how we can get from DNA to RNA and then from RNA to proteins, and it's a pretty cool animation. So micro students, click on that, check it out. It'll be worth your 4 minutes and 25 seconds. And the last thing we'll look at is ATP. Now, ATP is not a nucleic acid. However, it has a lot of the basic components of a nitrogenous base. It has ribose, it has adenine, but instead of one phosphate group, it has 3 phosphate groups, hence triphosphate. Adenosine triphosphate, ATP, it's the single most important energy transfer molecule in any biological system, in any cell. So these are, as you can see, the adenine joins with the ribose, the 5-carbon sugar, and then from the ribose we have the 3 phosphates. The energy is contained in the bonds of the phosphates, especially between the bond between phosphate 2 and phosphate 3. And when that bond breaks, energy gets released, that energy gets absorbed by the cell, and that energy drives all biochemical processes. So ATP loses a phosphate, becomes ADP, adenosine diphosphate, but then it'll pick up another phosphate, become re-energized, and then get broken down, energy gets released, and so on and so on. And this is how all of our cells survive. How is it made? Dehydration synthesis. Take ADP, adenosine diphosphate with 2 phosphates, add a phosphate and some energy that gets locked into that bond, and you have ATP and water leaves. It can be broken by hydrolysis to liberate the useful energy for the cell. So start with ATP, add water, and that's why we need to drink water. Water is becoming the basis of the chemical reaction to unlock the energy that's between phosphates 2 and 3, and then you end up with ADP plus a phosphate plus energy given off, and that energy then is used to drive the biochemical reactions within cells. And we'll talk about that in another chapter when we look at cell energetics. And we'll look at ATP a lot more. ATP becomes the star of the show. Well, that's it. That's the end of part 3 of this 3-part lecture on chemistry. Hope you enjoyed it, and hopefully was of some educational value to you. Thank you for watching, and if you find this interesting, I have plenty of other videos to watch on my channel called Tink Talks. And thank you for watching.